Hi, everyone. Thanks for being here today. I want to thank Rafael Cruz for having us invited to this symposium. My name is Natalia Pabon Mora. I am the principal investigator of the Plant Devo lab at the Universidad Antioquia in Medellin in Colombia. And today I would like to share this presentation with you, which I have titled A Plant Living Inside a Plant, Assessing Developmental Genetic Networks Under Extreme Holoparasitism. I hope you enjoy this talk. All right, so first I would like to tell you a little bit of our research in the Plant Devo group located in Medellin, Colombia. For 10 years now, we have been able to address different questions regarding the genetic mechanisms underlying the formation and elaboration of floral meristems, carpels, and fruits, the three key synapomorphies of all flowering plants. But because we're based in Colombia, a biodiversity hotspot, we have been able to study diverse groups focusing, of course, in non-model species. For instance, we have studied the genetic basis of fruit and pseudofruit formation in the Apodanthaceae, the genetic basis of floral organ symmetry, color, and epidermal elaboration in the Aristolochiaceae, the genetic control of flowering time in tropical orchids, the genetic basis of petal loss in tropical poppies, the genes linked to bilateral symmetry and spur formation in Tropiolum, and we have also tackled the genetic factors controlling fruit development in the Solanaceae and the Rubiaceae. And I am here today to tell you about the genomic content and some of the most remarkable developmental processes in miniature holoparasites in the Apodanthaceae. So I would like to introduce you first to the concept of holoparasitism. Holoparasitism has evolved at least 7 times independently during flowering plant evolution, and the group includes several fascinating organisms that have replaced their autotrophic ancestral condition and have become fully dependent on their host metabolism, acquiring both water and nutrients from another plant. For the rest of this presentation, I will focus on the Apodanthaceae, which you will see in the left side of the slide, a holoparasitic clade inside Cucurbitaceae. Now, the family Apodanthaceae comprises 2 genera and 12 species, all of which are holoparasites, while there is a single species in the genus Apodanthes in the left, which is found only in wet forests, parasitizing primarily Casearia, a member of the Salucaceae. The 11 species of Pilostyles, here to the right of the slide, are found in dry habitats, parasitizing non-closely related members of the Fabaceae. Clearly, there is an extreme habitat preference and a host specificity shift between the 2 genera. Something remarkable is that while Apodanthes is only found in the Americas, members of Pilostyles are found in 4 continents: Australia, the Near East, Africa, and the Americas. This intercontinental distribution of Pilostyles and the obligate legume dependence rule out long-distance dispersal, and instead support the idea of this conserved holoparasitic relationship being in place before the Gondwana split. In addition, if you look at the map to the left side, in our Andean Cordillera, the 2 genera can be found. Thus, being in Colombia has allowed us to sample the full representation of the family. So today I will present our ongoing research on this unique relationship between Pilostyles boyacensis, a species that we described in 2014, and you can see in the video its host, Dalea quatrecasaceae, both endemic from dry Andean valleys in Colombia. Again, what you're seeing here is the shrubby host plant being parasitized by numerous gregarious, presumably individuals of the parasitic Pilostyles. Interestingly, all members of the family are quite unique as they lack typical root and shoot apical meristems. They don't form any leaves and they lack any chlorophyll. In fact, most of their life cycle occurs inside the host, and they can only become evident when fully formed flowers emerge, breaking through the host bark, as you can see in this slide. Now, one of the most remarkable features of this endoholoparasite is that all stages of flower development occur completely inside of the host stem, starting from seemingly undifferentiated meristematic patches lacking any structural patterning, which are here highlighted in yellow in the first photograph, to fully formed flowers with all organs differentiated. Now, early stages of perianth initiation in the bottom portion of the slide also occur inside of the host in a spiral and acropetal sequence, from which the outermost ones will become purple and the inner ones will, in fact, remain white. Now, Pilostyles has unisexual flowers. The male flowers here in the left lack typical stamens. Instead, they have pollen sacs which mature from bottom to top, forming a cynandrium. Male flowers also have a highly elaborated central column, perhaps corresponding to a vestigial gynecium. The column here has vesicular hairs that mature concomitant to pollen maturation. The female flowers on the right side of the slide, on the other hand, lack any remnants of the male portions, and they're formed by four fused carpels. These flowers have a less elaborated stigma, a short style, but they carry numerous ovules, which after pollination, in fact, these female flowers form fleshy fruits, with numerous seeds apparently viable as they frequently have minute but completely formed embryos. In addition to staminate and carpellate flowers, we were able to record the rare occurrence of chimeric half-male and half-female flowers, in which the male half forms vesicular hairs and typical pollen sacs, and the female half forms seemingly normal ovules. Pollination, seed dispersal, and germination have not been well studied or studied at all. We have recorded the presence of abundant scale bugs that do seem to feed in the flowers as well as several ants passing through the flowers. Interestingly, the ants seem to be more interested in the fruits, and the seeds do carry eliosomes, which are tiny, oily bodies that are thought to attach to the ant. However, there is a clear need for more detailed ecological studies in this particular system. So such a unique system raises numerous questions. The first one is, how is the genome of this very strange plant? Is it reduced? What can we say about the likelihood of gene transfer between the parasite and the host? The second question is, which should apical meristem related genes are retained under extreme vegetative reduction in pilostyles? And the third question is, what is the developmental genetic basis for flowering and floral organ identity in pilostyles? So in order to answer these questions, we have generated genomic data as well as 3 different reference transcriptomes spanning the full life cycle of pilostyles. The first one corresponds to the endophytic tissue of pilostyles growing inside the Dahlia stem. The second one corresponds to pre-anthetic flowers already emerged, and the third corresponds to young and fully formed fruits with seeds. More recently, we have also generated control transcriptomes from the Dahlia healthy stems, lacking any trace of the parasite, and the Dahlia inflorescences. Now, even more recently, we began sequencing for Apodanthaceae, which was only found this year in the proper developmental stages. So the first and easiest to accomplish task was to sort out the contigs in order to identify genes belonging to different compartments, which was done based on depth and length. Now, this resulted in numerous contigs putatively assigned to the nuclear genome here highlighted in green, more abundant contigs currently being annotated as mitochondrial in pink, and a single super abundant contig corresponding to the plastid genome, which you can see here in the arrow. Now, the plastome in the New World Pilostyles boyacensis is almost 12,000 base pairs long, which is incredibly reduced compared to other flowering plants, but it is well within the range of previously annotated plastomes in the genus. Here, we were able to annotate 7 genes, including 2 ribosomal subunits, and 5 genes, including a putative oxidoreductase, which is the only gene that was actually not previously reported for any other Pilostyles species. We were also able to detect active transcription for all genes, suggesting that they are all functional, unlike previous reports of pseudogenization. And synteny maps show a large degree of positional concordance between all genes in the 3 plastomes sequenced so far. We are currently in the process of sorting out the genomic material in our metagenome for this symbiosis. Most of the parasitic nuclear and mitochondrial genomes are concentrated in this peak, which I have labeled peak 1 to the right. And we're currently assessing the mitochondrial genome, and I can already tell you that in this genetic compartment, there is some evidence of horizontal gene transfer. However, since our sampling is not axenic, we have found a second peak, which is here labeled as peak 2, likely belonging to the host plant, as well as peripheral bacterial and fungal genomes, putatively taking part in this system as well. Okay, so now let me move forward to the more developmental part of this story, our work on the genetic mechanisms underlying shoot apical meristem development. And in fact, I want to start by reminding you that the basic body plan in vascular plants results from the activity of 2 meristems, the root apical meristem developing the underground system, and the shoot apical meristem, which, as you know, is responsible for the formation of all aerial organs post-embryonically. It is, in fact, that shoot apical meristem, the one that forms the leaves and eventually suffers a transition to an inflorescence meristem during the reproductive phase. So, several genes are known to control apical meristem development, and they have been largely identified in Arabidopsis thaliana. The most important transcription factor involved in promoting stem cell maintenance is WUSCHEL. WUSCHEL is expressed in the organizing center, but its protein can move towards the central zone. If the activity of WUSCHEL is affected, the meristem stops proliferating and it depletes completely. Interestingly, the same WUSCHEL activates its counteracting clavata factors. First, clavata 3, which is activated at the beginning. It encodes a small peptide that can be secreted into the extracellular spaces in the central zone. And then its receptor follows, which is clavata 1. Clavata 1 is turned on, and it's actually the interaction of clavata 3 and clavata 1 which triggers negative regulation of WUSCHEL, allowing cell differentiation and lateral organ formation to occur in the periphery of the meristem. Now, this finally regulated feedback loop is reinforced by two additional factors: BAM receptors. BAM stands for barely any meristem, and they are in charge of capturing any remnant clavata-3 peptides. And on the other hand, WOX9. WOX9 is a homolog of WUSCHEL, which is in charge of promoting pluripotency. So our goal in this context was to isolate all homologs of these genes from Pilostyles, which in fact belong to 3 different gene families, in order to test which of these genes were still present despite the extreme reduction of the shoot apical meristem of Pilostyles. And what we found was quite remarkable, as our results showed that Pilostyles has active WUSCHEL homologs, but surprisingly, they are not active in the earliest developmental stages, suggesting that their role is late in flower development. Instead, and perhaps in replacement of early WUSCHEL activity, we did find WOX9 in all 3 samples, spanning the full life cycle of Pilostyles. And in fact, we tested the expression of WOX9 by in situ hybridization and found that this is a gene already expressed in embryos, even in the seed coat, possibly also in the endosperm, and the same gene stays active also during the development of the endophyte. Surprisingly, we were not able to find any active homologs of clavata3 or clavata1 in any of the developmental stages sampled. And finally, we were able to identify BAM receptors. However, as their role is to quench out the excess of clavata3, it is unclear what their role is in this system lacking clavata3 homologs. Okay, so at this point, I can provide 3 conclusions. The first one is that Pilostyles shows at least 1 transcript with all functional domains conserved for BAM1, WUSCHEL, and WOX9. The second is that there are no clavata3 or clavata1 homologs in Pilostyles, and this means that there are either alternative repression modules for pluripotency, or that no repression is needed as there are no lateral organs being formed. And finally, that the minimal genetic regulatory network for pluripotency regulation in this holoparasite requires only WOX9 and BAM1, as they are the only genes present in the endophyte. All right, so what about the genes involved in flowering and floral organ identity? Well, it turns out that despite the extreme reduction of the vegetative phase, Pilostyles has intact flowering and floral organ identity. Okay, so briefly, I want to summarize what we know from the genetic control of flowering. At least in the model species Arabidopsis thaliana, flowering is controlled by the age of the plant, the hormonal signals, light, and temperature. It all really starts with FT, which is a mobile signal produced in the leaves that triggers phase transition, activating 3 additional factors: AGL24, SOC1, and finally LEAFY, which controls floral meristem identity, here shown in the yellow stars to the right. Now, these factors are known as the positive flowering integrators. If any of these genes is mutated, plants can transition to the floral phase. On the other hand, there are the negative flowering regulators, which include FLC, SBP, and TFL1. Mutation of these genes results in early flowering. Now, these genes all belong to 3 very different large gene families. And interestingly, closely related paralogs belonging to the same gene family can have totally opposite functions during that flowering transition. Okay, so our goal was to identify putative homologs of these genes in Pilostyles, and our results so far from the phylogenetic reconstructions of the PDP, the AGL24SVP, and the SOC1 gene lineages suggest that all flowering integrators in this system belong to the host, Dalea quatrecasaceae, as all genes identified cluster with Pabon homologs, which are indicated with stars in all three phylogenetic reconstructions, with the exception of one single SOC homolog, which you can see in the right portion of the slide. Remarkably, these results suggest that Pilostyles recruits the flowering signal from its host, which is not unusual, as the same trend has been reported in the genus Cuscuta, for example, the daughters. So in an attempt to get an overview of how much of the flowering signal can be recruited from the host, we have blasted all flowering-related genes in photoperiod, vernalization, the autonomous pathway, and the central flowering integrators. And basically everything you see in green likely belongs to the host plant, and everything you see in blue or gray, we have not been able to identify from the transcriptomic data. And only really these two genes in purple are those that undoubtedly belong to the parasitic plant. Okay, so conversely, leafy, which is the most important gene for floral meristem identity, is endogenous, meaning it is expressed by Pilostyles and not by its host. Using in situ hybridization analysis, which you can see in this slide, we have been able to track leafy expression very early on, starting from the meristematic endophytic tissue that now we know is extremely unpatterned floral tissue, all the way to fully formed and really basically emerged flowers. So this is quite exciting, as leafy can be used then as a marker for Pilostyles differentiation inside of the host. Finally, we wanted to assess the genes involved in floral organ identity, and recall that those genes are members of the Matchbox transcription factor family. The model established in Abrasis based on the homeotic mutants you see in this slide points to 4 different classes of transcription factors, the A, the B, the C, and the E class genes, whose overlapping expression controls the identity of all floral organs. So what we were able to find is that all floral organ identity genes in Pilostyles, which as leafy, they're likely endogenous in the parasite. All endogenous genes are pointed with arrows in this slide. However, a few homologs cluster with Pabon homologs indicated with the stars. We are still exploring what that means, but we think that all homologs that cluster with Pabon in fact belong to Dalea and are just broadly expressed in the stem. So far, all the core floral organ identity genes have been found in Pilostyles. Okay, so this leads me to the following conclusions. The first one is that the floral promoters, FT, AGL24, and SOC1, likely belong to the host, but are being used by Pilostyles. The only flowering repressor identified so far, SVP, also belongs to the host. Conversely, floral meristem identity genes and most floral organ identity genes are endogenous in Pilostyles. And finally, we are happy to report that leafy expression marks floral meristem fate, even during very early cryptic development in Pilostyles. All right, so with that, I would like to thank the Universidad Nacional de Colombia for funding this research and all the people that have been involved in different stages of this project, especially my co-authors on this talk, which you will see pointed with the arrows, Fabio Gonzalez in the top of the slide, Angie Daniela Gonzalez, bottom part of the slide, and Juan Fernando Alzate, right part of the slide, and all the undergraduate and graduate students that have participated in the project. And I want to thank you for your attention, and I will be happy to answer any questions.